Welcome back. So this is the third video. What we've covered so far is transcription in both prokaryotes and eukaryotes. Now after transcription in prokaryotes, mRNA is ready to go. But in eukaryotes, we had to do some RNA processing before we were ready. But now we have our completed strand of mRNA and we're ready for the next step, which is translation. And this is the process of RNA being translated into proteins, changing that nucleic acid language into the protein language. So just like with transcription, we're going to explain it by breaking it up into the major players first. With transcription, we talked about RNA polymerase and promoters first before talking about the actual process. We're going to do the same exact thing with translation. So mRNA needs to move, at least in eukaryotes. In prokaryotes, both processes happen in the cytoplasm. But in eukaryotes, it doesn't happen that way. Transcription was in the nucleus, which is where the DNA is, and then it's going to move from the nucleus to the cytoplasm. So it's a very key difference between prokaryotes and eukaryotes. And you can kind of see that in this image. Here we have RNA. You see tons and tons of it in the nucleus. Give it about an hour to complete transcription, and now you see this RNA all over the cell. But again, in prokaryotes, there's no nucleus, so there's no movement. So an overview of translation. Here we had DNA. It was transcribed into mRNA, and then this gets translated into proteins. But we're still kind of missing a key step. How exactly are we translating mRNA into proteins? How does an amino acid associate with mRNA? Amino acids don't have nitrogen bases to bind with mRNA. So the question is what are we missing? So in this diagram, also an overview of translation. Here's the ribosome. Go mRNA. Here's the amino acids, but you also notice we have a middleman. We have a go-between between the nucleic acid language and the protein language. And that molecule, which we'll discuss more, is tRNA. So think of T as transfer. This RNA is going to be transferring these amino acids to that mRNA and really reading those mRNA instructions in order to make this long polypeptide chain or this really long protein. So tRNA or transfer RNA. Here's a picture of what it looks like. We think of RNA as being single-stranded, and tRNA is single-stranded, but it has some secondary and some tertiary structure. It folds in on itself. So DNA was double-stranded, two separate strands, and although it kind of looks like that with tRNA, that is not the case. It is one strand. It's just folded up in and of itself. There's some alpha helices, there's some beta sheets. So it does have secondary and tertiary structure. And its whole goal is to pick up an amino acid. You can see that bound here at the end of this RNA molecule, this tRNA molecule. In this case, we have SER, which is serine. And on the other end, it has this recognition site where it's going to recognize what mRNA has been saying. So for instance, mRNA has a codon that says UCA. Well, tRNA has the complement to that. In this case, it has AGU. It's complementing that mRNA, because remember, if mRNA says U, another U doesn't fit onto that. A G or a C won't fit onto that. Only an A or an adenine will fit onto a uracil. So think of this area, which is called the anticodon. Think of that area as just a way to temporarily bind with mRNA, kind of ensuring a sure fit. Now this anticodon, so thinking of the codon on mRNA and then the anti or the opposite on tRNA, you notice it's running 3' to 5'. So here's the 3' labeled up here. We can follow this strand all the way down. And so here's 3' here and then 5' here. And that should make sense because this mRNA strand is labeled 5' to 3'. Well, when you do this matching, this base pairing, you have to be anti-parallel. It's the same thing we saw with DNA synthesis. It's the same thing we saw with RNA, mRNA being transcribed. So the anticodon runs the same way. It runs 3' to 5' in order to complement the 5' to 3' of RNA. This is more of a digital structure of what tRNA actually looks like. So tRNA, when it has its amino acid, because tRNA will lose those amino acids every time it undergoes translation, because it's going to drop off those amino acids to that growing protein. So when tRNA has its amino acid attached to it, we call that whole structure an amino acyl tRNA. So it's still a tRNA. Everything about tRNA is the same. It just has its amino acid on it, and that's what this little purple pink area is representing. So this whole molecule we would call the amino acyl tRNA. Now I keep highlighting this green. What that green is an amino acyl tRNA synthase. This is an enzyme. So this green area right here, this is all an enzyme. The responsibility of this enzyme is kind of in the name. It synthesizes amino acyl tRNAs. The whole job of this enzyme is to put that amino acid on the appropriate tRNA. Now tRNA can only hold one amino acid, and it's actually very unique. So one type of tRNA, the one that has that anticodon of AAA, is always going to hold the same exact amino acid. So the role of these enzymes is to put those amino acids onto tRNA. Hopefully you remember there's 20 different amino acids, so it should be no surprise that we have 20 different amino acyl tRNA synthases. Because you have 20 amino acids, so you need 20 enzymes to put those 20 different amino acids onto tRNA. So these enzymes are incredibly crucial. Although they don't play a direct role in protein synthesis, without them, none of our tRNA would have their amino acids. They would drop off their amino acids during translation, and then that's it. Now this process, it is a synthesis process. And whenever you're going to bring two things together, that requires ATP. And this process is no different. So just kind of as a reminder, this is a synthesis reaction. We're not using, DNTPs or anything like that. This is a process that will require energy in order to complete. So this is one of the things that our ATP goes to in our cells. You'll see tRNA drawn a couple different ways, but they all have the same features. Here on the left is probably kind of a more flattened shape, looking at how tRNA looks just looking at the sequences. And again, you do see it is single-stranded. It's just folding back in on itself. At one end, this is where the amino acid will attach, and on the other end is the anticodon. In this case, it's AAG. So if it's AAG, let me bring up my pointer. So I have a question for you. If it's AAG, what mRNA codon will it bind to? So hopefully your answer, so adenine is going to bind with uracil, adenine is going to bind with uracil, and guanine is going to bind with cytosine. So this particular tRNA molecule is going to attach to an mRNA codon that reads UUC. And then whatever amino acid's hanging out here will go along with it. This is the kind of structure we were looking at really but a slide ago. Excuse me. And then this is just a super simplified version of it, but all three of these representing the same thing. That middleman that's going to bring that amino acid to the ribosome and copy or read the directions mRNA says. So if you look at this genetic code, remember this codon table. We visited this in chapter 16 when investigating the central dogma. How many codons do you see? Remember, this is all mRNA. How many codons are there? So you could count each of these individually, but just to shorthand it, there's 16 in each of these columns, and there's 4 across. So that actually equals 64. So there's 64 different combinations of codons, of all of these 3 different nucleotide spots. So it would stand to reason that if there's 64 codons, well, that means you need 64 anticodons to account for every single type of codon you might interact with. So you would say 64. But, you know, we have a stop codon, and a stop codon. Those don't code for anything. There's no tRNA for those. They don't carry an amino acid. That's just a little section that says, hey, this is it. So technically, we would expect 61 because those codons don't actually code for anything in tRNA. So given this information, you would expect 61 different tRNAs. But in actuality, we have 40 different types. But how can that be? You know, all 64, or really 61 of these, has an equal likelihood of showing up in mRNA. So what happens when you don't have the right tRNA? Maybe this UCU, maybe that tRNA that codes for that is missing. Maybe we don't have that. Well, what happens if we come across that in mRNA? So our cells have addressed this. It's called the Wobble Hypothesis. And what that means is that one tRNA can actually base pair with more than one type of codon. And that kind of flexibility is what we refer to when we say wobble. So let me give you an example, thinking about that third position of the anticodon. Remember when we were talking about silent mutations, that if that third nucleotide in a codon was a different letter, that sometimes it would code for the same exact amino acid. Well, the same is true with tRNAs. So let me give you an example. We have 2 different codons here, coding from mRNA. We would expect tRNA of UGA, we would expect, I'm going to write T here, we would expect to say A, C, U. And if it came across this codon over here, we would expect it to say A, C, C. So 2 different tRNAs would be needed. One with an anticodon that says ACU, one with an anticodon that says ACC. But in actuality, our cells only actually have one type of tRNA. It doesn't have an ACU and an ACC. So the Wobble hypothesis has shown that, hey, right here we do have a perfect match. tRNA should say ACU, and hey, it does say ACU. But this same exact tRNA that says ACU can actually code this one as well. It should say ACC, but there's a little bit of wiggle room. There's a little bit of wobble where this tRNA could still attach to it, even though it's not a 100% perfect match. Which is really cool because that's why we can have 40 tRNA molecules, even though we have 61 different codons. Here's another example. So here you have an mRNA that says CUC. Here's one that says CUU. But actually, the same tRNA can bring leucine. The GAG tRNA will bind to both of them. We see this the most between U's and G's. That's the one that has the most wobble. That a U and G can kind of pair, you know, G and C usually pair. But U and G's can kind of pair. So that's where the most wobbling happens. So we're going to end it there for now. Your next video is going to cover ribosomes and begin the translation process.